हेलो 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 हेलो। वॉइस आ रही है मेरी? स्टार्ट होगी स्ट्रीम। एक सेकंड में वॉइस चेक कर लेता हूँ एक बार। हेलो हेलो। चलो वॉइस सही है। हाय गाइस, वेलकम टू माइक्रोएक्स चैनल। और आज मैं आपका न्यू सेशन लेने वाला हूँ पीटी का 20 मॉड्यूल। आज हम 20 मॉड्यूल कवर करने वाले हैं, राइटिंग का, रीडिंग का, स्पीकिंग एंड लिसनिंग ऑल्सो। तो देखते हैं कहाँ से स्टार्ट करते हैं। पहले तो हम इम्पोर्टेंट सेशंस देखेंगे, इम्पोर्टेंट मॉड्यूल कौन कौन से हैं, वहीं से स्टार्ट करते हैं। चल मैं स्क्रीन शेयर करता हूँ आपसे। चलो आज देखते हैं हम कहाँ से स्टार्ट करते हैं। पहले हम टाइमर लगाएंगे 1 घंटा 1 घंटा 1 घंटा 1 घंटा करके, वन 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 हावर करके। पहले मैं सोच रहा हूँ कि हम हाईलाइट इनकरेक्ट वर्ड्स करते हैं, लिसनिंग से स्टार्ट करते हैं, क्योंकि शुरू से ही हम पहले राइटिंग से स्टार्ट करते हैं, आज लिसनिंग से स्टार्ट करते हैं। हाईलाइट इनकरेक्ट वर्ड हम करने वाले हैं 20।
What we're going to find out today is how it's a bit more complicated. What we're going to find.
मैं 1 घंटे का टाइम लगा देता हूँ। ट्वेंटी हम हाईलाइट्स करेक्ट वर्ड करने वाले हैं। उसके बाद राइट फ्रॉम डिक्टेशन करेंगे। पहले लिसनिंग पार्ट को थोड़ा कवर कर लेते हैं। आज मैं थोड़ा क्या कहते हैं मिक्सिंग करूँगा, कभी कुछ कभी कुछ। तो स्टार्ट करते हैं। वॉइस अच्छे से आ रही है ना, एक बार बता दो मेरे को। आज लिसनिंग पार्ट कवर करते हैं। अब देखते हैं ये कैसे होते हैं। देखो,
What we're going to find out today is how it's a bit more complicated than that. What we're going to find out today is how it's a bit more complicated than that, which it always is. And I think it's really wonderful. I mean, not being an experimental scientist myself, I have a kind of envy at the way in which science can continue to surprise us by this. People working away in labs, moving on understanding in ways. Hugo is a cognitive scientist at the French National Centre for Scientific Research. Hugo Mercier.
ये देखो, अब देखते हैं मेरा क्या बना है। 5 × 5, ठीक है। तो ये इसमें हम क्या करना होता है, इसमें मेनली ये स्पीकर लेक्चर देगा, उसमें से हमें वर्ड्स को इनकरेक्ट करना होता है, लाइक पकड़ना होता है कौन कौन से वर्ड्स इनकरेक्ट है। उसको बहुत स्पीड तेज भी हो सकती है एंड लोअर भी हो सकती है। तो स्टार्ट करते हैं। वेलकम विजय भाई, वेलकम नवीन भाई, वेलकम टू माइक्रो एक्स। वेलकम वेलकम। नेक्स्ट कर रहे हैं। अब एक बार टाइमर ऑन कर रहा हूँ। हम ट्राई करते हैं।
The world has changed. The economics of the world have changed, and the art market has come in.
एक मिनट मैं ट्राई करता हूँ। 20 हम करेंगे, शॉर्ट टर्म डेडलाइन यूज़ करने का ट्राई करेंगे कि 30 मिनट्स के अंदर हम 20 करेंगे। वैसे नहीं हो पाएंगे, लेकिन इससे क्या होगा कि हमारी स्पीड भी बनी रहेगी और हम काफ़ी सारे मॉडल को इसलिए जल्दी कवर कर सकते हैं। तो 30 मिनट्स के अंदर आप फोकस के साथ हम सेशन को स्टार्ट करते हैं। स्टार्ट करते हैं. ओके
The
रुको.
world has changed. The
आई थिंक ये प्रैक्टिस्ड है. अनप्रैक्टिस वाले निकालता हूँ. अनप्रैक्टिस एंड इसको मीडिया.
Those clever little monkeys are on strike again, and I don't mean the Writers Guild. No, I'm talking about the tufted capuchin monkeys at the Yerkes Primate Research Center. Those headstrong primates have put down their feet, all 4 of them, and refuse to work for unfair wages. Their grievances are presented in the current online edition of the Proceedings of the National Academy of Sciences. To examine Capuchin's sense of fairness, Franz de Waal and his colleagues at Emory University designed a bartering task, in which monkeys are given tokens they can then exchange for food. Some monkeys get a plain old cucumber slice, others get a coveted grape. What the researchers discovered is that when monkeys who get salad fixins see their friends get fruit, they basically refuse to play anymore. But are the monkeys really reacting to inequity, thinking, that's not fair, she got a grape and I didn't? Or are they just being greedy? I see grapes, I want one. Or maybe they're frustrated. Last time I got a grape, now this? The current study suggests that the grapeless monkeys indeed consider themselves shortchanged. Tests to see what they might type, given an infinite amount of time, are on hold until both labor disputes are settled.
आई थिंक इसमें मैंने एक छोड़ दिया है. देखते कितने शायद से एक छूट गया. कहा था मैंने क्योंकि 4 है इसमें से. ये मैं इसमें कंफ्यूज था एक दो में. नींद आती.
Back in ancient times, philosophers like Aristotle were already speculating about the origins of taste and how the tongue sent.
नाउ स्टार्ट. चाय पी ले, अब देखते हैं.
Back in ancient times, philosophers like Aristotle were already speculating about the origins of.
चाय मैं तो ऑन नहीं किया था.
Back in ancient times, philosophers like Aristotle were already speculating about the origins of taste and how the tongue sensed elemental tastes like sweet and bitter, salty and sour.
What we discovered just a few years ago is that there are regions of the brain, regions of the cortex, where particular fields of neurons represent these different tastes again. So there's a sweet field, a bitter field, a salty field, etc.
Nick Reba, a sensory neuroscientist at the National Institutes of Health. Reba and his colleagues found that you can actually taste without a tongue at all, simply by stimulating the taste part of the brain, the insular cortex. They ran the experiment in mice with a special sort of brain implant, a fiber optic cable that turns neurons on with a pulse of laser light. And by switching on the bitter sensing part of the brain, they were able to make mice pucker up as if they were tasting something bitter, even though absolutely nothing bitter was touching the tongues of the mice. In another experiment, the researchers fed the mice a bitter flavoring on their tongues, but then made it more palatable by switching on the sweet zone in the brain.
What we were doing here was adding the sweetness, but only adding it in the brain, not in what we were giving to the mouth.
Think adding sugar to your coffee, but doing it only in your mind. The results appear in the journal Nature. Reba says the study suggests that a lot of our basic judgments about taste, sweet means good, bitter means bad, are actually hardwired at the level of the brain. And as for that virtual sugar in your coffee idea,
I think it's basically science fiction to think that this would be something that would be applied to humans.
But today's science fiction might be tomorrow's artificial sweetener.
मेरे को वॉइस आ रही है, एक बार क्लियर करना. मेरी वॉइस आ रही है और स्क्रीन में जो चल रहा है लेक्चर उसकी वॉइस आ रही है. एक बार क्लियर करना कि वॉइस आ रही है कि नहीं अच्छे से. Okay.
This is Scientific American's 60-Second Science. I'm Karan Hopkin. This will just take a minute. As kids, many of us pondered what stars are made of. Some grown-up astronomers, on the other hand, wonder about where stars came from. Now, a study serves up a surprise, because it seems that the smallest galaxies in the universe gave rise to an unexpectedly large proportion of stars. The findings are in the Astrophysical Journal. Most of the stars we see in the sky were formed when the universe was young, just a few billion years after the Big Bang. So, to study stellar origins, scientists use telescopes that allow them to see galaxies that are so far away, they're essentially looking back in time. Previous observations had focused on the star-forming powers of larger galaxies. But in this latest study, researchers used data collected by a powerful camera aboard the Hubble Space Telescope. With this instrument, they could eyeball smaller, dwarf galaxies, and they found that these diminutive dynamos churned out stars at a furious rate, fast enough to double their mass in only 150 million years. That reproductive feat would take most so-called normal galaxies 1 to 3 billion years. Seems the universe has long known what Danny DeVito, Michael J. Fox, and Dustin Hoffman later proved. You don't have to be big to have real star power. Thanks for the minute. For Scientific American's 60-Second Science, I'm Karan Hopkin.
एक सॉल्व हो गया. ये इसमें मेरे 2 रहेगी, कोई बात नहीं, नेक्स्ट करते हैं हम. एक तो मैं तो कोई प्रॉब्लम ही नहीं होता, और वैसे भी नहीं तो इसे कम ही होता.
This is Scientific American's 60-Second Science. I'm Molly Webster. Got a minute? The element mercury has been shown to damage the human nervous system. Yet, since 1856, mercury amalgam is the filling dentists have used to repair our teeth. Now, a study in the journal Chemical Research and Toxicology indicates that mercury fillings actually lose their toxic potential over time. Canadian scientists use X-ray spectroscopy to compare new mercury amalgams to those about 20 years old. After 2 decades, 95% of the elemental mercury on the surface of a filling had gone through a chemical reaction. It became beta mercury sulfide, a black-colored solid, which explains why fillings dull with age. And the compound isn't absorbed by the body, which means it can't affect the body the way elemental mercury can. Of course, this study does not settle the question of how potentially harmful a new mercury filling is, but sulfide-rich foods such as coffee, garlic, and onions may accelerate the change from metallic mercury into the safer sulfide. So if you have a new mercury filling, an odoriferous chili dog with a cup of coffee may be just what the doctor ordered. Thanks for the minute. For Scientific American's 60-Second Science, I'm Molly Webster.
देखो, अब इसमें मेरी नेगेटिव मार्किंग भी होती है. अब इसमें मेरे 5 सही थे, 1 2 3 4 5 नहीं 6 सही थे. 6 में से मेरे क्या हो गया, एक की तो मार्क्स ही कट गया, एक ये छूट गया, 2 छूट गया, 3 कट गया, 2 छूट गया और एक गलत होने का नेगेटिव हो गया. 5 में से 4 इसलिए हो गया क्योंकि एक नेगेटिव में चला गया, तो इसमें नेगेटिव हो जाता है. तो जिस चीज़ में कंफर्मेशन नहीं वो छोड़ दो, नहीं तो भाई मार्क्स ही जाने हैं. अब नेक्स्ट करते हैं हम, और भी प्रैक्टिस में मैटर करती है.
This is Scientific American's 60-Second Science. I'm Karan Hopkin. This will just take a minute. Since the first human genome sequence was published, thousands of people have submitted their DNA for scientific analysis. They made these donations anonymously, or so they thought. Now, using publicly available information, researchers found they could figure out the identities of 50 individuals who had loaned their genes to science. Their results, although not the names of the people, are in the journal Science. Biomedical research depends on human subjects, and issues of privacy have always been a concern. When scientists share genomic data, they first strip away identifying information, like the individual's name and date of birth. But is that really enough? Researchers looked at a specific set of markers in genomes whose sequences were in a public database, and they found that by matching up these markers with sequences that people had submitted to genealogy websites, they could identify some of the genome donors' relatives, and, with a bit more sleuthing, come up with their actual names. Of course, many people now post online accounts of what's on their minds or even on their menus. But even those who are relatively relaxed about their privacy might think twice about their genomes going public.
इसमें
Thanks
से
for the minute.
एक
For
बार
Scientific American's 60-Second Science, I'm Karan Hopkin.
मैंने बोला था इसमें से एक तो छूट गया, इससे आगे ना ये मैं कंफ्यूज था ये अगर इसको क्लिक कर देता तो नेगेटिव भी चला जा सकता था, इसे मैंने छोड़ दिया। चलो कम से कम आगे आके करते हैं।
If you've ever been to an all-you-can-eat buffet, you know how important it is to carefully weigh your options.
फिर से बोलो।
If you've ever been to an all-you-can-eat buffet, you know how important it is to carefully weigh your options. You don't want to fill up on salad when so many calorie-laden delights await. It seems some birds also weigh their mealtime choices, literally. A study finds that Mexican jays pick up and shake peanuts to assess their relative heft before choosing one. That report is served up in the Journal of Ornithology. Foods that hide their edible bits on the inside present a challenge to hungry diners. How can you tell which fruits are ripe or which shells harbor the biggest nuts? We humans knock on melons or squeeze avocados, but how do other species select the highest quality snacks? To find out how the jays do it, researchers fiddled with their feed. First, they doctored peanuts so that some contained 3 nuts while others had none. When they offered these pods to some jays, the birds turned their beaks up at the empty shells and instead chose those that were full. And when the jays were allowed to choose between normal peanuts and those that weighed just 1 gram more, because the researchers had stuffed them with clay, the birds again went for the heavier meal. Videos revealed that the jays shake the nuts before selecting one, which apparently lets them gauge the nut's mass and perhaps also listen for the rattle of a well-packed shell. Pretty clever for a bird brain.
लिख रह गया था, बाकी सारे सही हैं।
When is an ant like a piece of fruit?
कैसा लग रहा है दोस्तों बताओ। नेक्स्ट का क्या है? तो छूट ही रहे हैं इसमें से। एक सेकेंड।
if
हेलो हेलो, मेरी वॉइस आ रही है? स्क्रीन की वॉइस सुनाई नहीं दे रही, आई थिंक, है ना?
Marmoset. They both like a little. What does a bookworm have in common with a black tufted marmoset? They both. What does a bookworm have in common with a black tufted marmoset? They both like a little quiet. Or so say scientists in the Royal Society journal Biology Letters. As urban areas continue to expand, their human inhabitants spread all sorts of pollution: air pollution, light pollution, even noise pollution. Each of these environmental encroachments affects the survival and behavior of local wildlife. Monkeys are no exception. Scientists in Brazil were studying how marmosets in a city park cope with ambient noise, and they found that the monkeys tend to settle down in whatever part of the park is most quiet. On weekdays, the marmosets steered clear of the roar of traffic, and on Sundays they shunned the clamor of visiting crowds. They dodged all the din, even when moving meant that they had to leave behind the places that offered them the most food. Could be that the monkeys are just skittish when it comes to unwelcome sounds, or maybe they can't hear what their friends are saying when it gets too loud. Either way, next time you see a cute little marmoset, forget ooh or aw or ee. Try shh.
एक तो मम्मी आ गई थी, डिस्टर्बेंस कर दिया।
Shh, keep this podcast a secret because new research points to a possible blind spot in airport security screening. It may be easier to sneak something dangerous past security, a box cutter, for example, by also including an obvious and innocuous banned object like a water bottle into the mix as a distraction. Scientists recruited college students to find targets on a computer display. Their task: search for lines that formed a T amidst other non-T lines in 10 different experiments. Sometimes the T's were easy to find, sometimes they were more hidden. When the easy and tough ones appeared with equal frequency, the students found both on the same screen. When the easy T's appeared 2 to 3 times more frequently, the students were more likely to miss the tough ones. But when students were given extra time, lessening the pressure, they were more able to find both targets almost as quickly. The study suggests that actual professional screeners need to be just as vigilant in their attention after finding a first piece of contraband in a given bag, and keeping their stress levels as low as possible should help their performance.
मिक्स करते हैं.
Creationists often publish lists of a few dozen scientists who doubt Darwin. So in 2003, the National Center for Science Education put together a list of 200 scientists who accept evolution.
Except that all of ours were named Steve.
That was the NCSE's Eugenie Scott at last week's AAAS meeting.
And now we have 1,000 scientists named Steve. Project Steve has a serious message. Approximately 1% of Americans are named Steve or Stephanie, so do the math. Our 1,000 Steves represents 100,000 scientists accepting evolution, as opposed to the rather paltry number dissing Darwin. It's important because in states where we have major problems with anti-evolution going on, the number of scientists doubting evolution has been proclaimed to the public. I just want the press to keep asking, how many Steves do you have?
And the 1,000th Steve is
the distinguished botanist at the University of Tulane and head of the Tulane Herbarium, Steve Darwin.
Chameleons have it easy. If they need to hide from a predator, they just change their appearance and disappear against whatever they happen to be standing on. Surrounded by leaves, turn green. Tree trunk, brown it is. But Aegean wall lizards have a different strategy. They live on the Greek islands, and instead of changing their appearance, they find a safer spot. That is, they're really good at picking out just the right background to minimize the chances that they'll be spotted by a hungry crow or raptor. That's according to a new study in the journal Scientific Reports. Birds can perceive both visible and ultraviolet light, so researchers snapped regular and UV photos of lizards basking on rocks to figure out how the lizards look to birds. The scientists discovered that the lizard's camouflage is precisely tuned to avian eyeballs, helping them seem to disappear. Lizards with darker backs hang out on darker rocks. Lighter ones use lighter rocks. It's the first time that wild animals have been recorded the best background to enhance their own camouflage. Another observation: when lizards aren't under the constant threat of death, they're less picky.
On with the lower risk, this kind of background choice to improve their level of camouflage is much less evident than on the islands with high predation risk.
University of Cambridge zoologist Kate Marshall, lead researcher of the study. To pick out the right rocks, Aegean wall lizards must have an awareness of what color they are. But how do they know?
Yeah, it's a big puzzle, and I'm fascinated by the answer. Unfortunately, I don't know the answer yet, and hopefully this study will inspire future research into it.
Mm-hmm.
Have a dog, you know you gotta walk it. But do you know how it walks? Well, if you have no idea which foot Fido puts forward when, you're in good company. Because according to a study published in the journal Current Biology, even places like natural history museums get it wrong half the time. Studies published back in the late 1800s showed that all four-legged animals walk the same way. They start by moving forward their left hind leg, followed by the left front leg. Then they repeat the sequence on the right-hand side. Different animals differ in the timing of their steps. The reason they walk that way is for stability. Lifting one leg at a time leaves 3 feet on the ground, forming a nice stable tripod to stand on. But not everyone seems to know that, even folks who should. Scientists looked at 300 depictions of animals walking, in museums, anatomy textbooks, even children's toys, and they found that nearly half the time these images get it wrong. For a toy, that kind of inattention to detail might mean that Rex has a tendency to roll over. But for museums to mess up like that, they just don't have a leg to stand on.